Okay, welcome back. We are going to be doing Lab 5, so we're going to be taking everything that we've learned before with how to collimate a light source, how to evenly illuminate a sample, how to make an infinity corrected system, and how to use a digital camera in order to create sort of the complete setup of a digital Köhler microscope. So what I have already done is I already have a nice collimated light source. The LED is focusing to a very far distance away. So we are going to be incorporating the iris. We're going to have a condenser lens for the first time. We have our sample holder. We have our objective lens and our tube lens that we used last time, and we also have our camera. So I'm going to be assembling this microscope in this order. We are going to be able to move this iris back and forth to sample different focal planes. So I will assemble these components and then we will get everything aligned. Now, because we have a curved surface on our condenser lens, one side curved, one side flat, we will need to make sure that we put the curved side towards the collimated rays, which these should be relatively collimated. Next, we're going to have our sample plane that we can mount slides and rulers in. We have our objective lens. Again, curved goes to collimated, so we're going to have divergent rays coming off of our sample, and it will be collimated behind it, so make sure to put the curved side back there. That also allows the objective lens to get nice and close to our sample, which is useful for an objective lens that has such a short focal length, in this case 20 millimeters. Now, you should have all of these focal lengths recorded from Lab 2, so go ahead and pull those up. Those will be necessary for later in the lab. Next, we have our tube lens, which we actually calibrated the magnification on last time in conjunction with this objective lens. So we will be sliding that on. And then finally, we have our camera. I'm going to go ahead and uncap my camera. There shouldn't be anything in there in the plane, but hopefully there will be soon. Okay, so we need to start getting this aligned. Well, so we will leave the iris for later. We'll just sort of leave that alone for right now and just start to consider what do we need for these components. So we know that our condenser focal length is 35 millimeters, and our sample plane is here. So first things first, we need to make sure that our condenser lens is about 35 millimeters away from our sample. Right. And that looks about good to me, but one thing that I will check is just to make sure that we have a nice focused beam of light at that plane, because again, that's what the condenser does, is it delivers our rays from our collimated LED onto our sample plane so that it's nice and even. That looks good to me. So we have our sample plane mounted one focal length away from our condenser. Next, we're going to need to put our objective lens one focal length away from our sample, because it will be taking any divergent light and collimating it away. So I'm going to mount this 20 millimeters away. I'm going to lock these down, but not permanently, because we will sort of need to slide them and adjust as we make the final adjustments. All right, so objective lens is roughly 20 millimeters away. Now we know that the objective lens is going to have a back focal point or back focal plane, which is going to be roughly 20 millimeters away from that. And you can probably visualize that focusing. I'm going to just do a quick measurement so that I mark pretty much where I expect that back focal plane to be. And then we have our tube lens. You should already sort of know what magnification this will produce. Actually, now that I think about it, I believe that we calibrated the magnification for no, we did it. We did it with this tube lens. So this guy has a focal length of 70 millimeters. So we're going to make sure that from this back focal point, this is mounted 70 millimeters away. So that's going to be about here. And I will lock that down. Again, if we had used the 300 millimeter focal length one, it's going to need to be mounted a lot farther back. But for this lab, we're just going to be using the 70 millimeter focal length lens. Now, we know that our image plane should be one focal length away from our tube lens. And that's, again, another 70 millimeters. So we're going to slide our camera down in there. So we should be close to somewhat of a infinity corrected Köhler microscope setup, but obviously we're not seeing anything on our image plane because we don't have a sample mounted. So the best thing that we can do is go ahead and mount a sample. I'm not going to be using the ruler today. I'm going to be using this calibration slide that we used in the last lab. Not for any sort of calibration because we should know what the magnification is, just to get a nice sharp image in our focal plane. So I'm just going to slide that in there and just going to be sort of looking for where anything starts to appear in my field of view. And I'm starting to see something there. So it looks like we've got a dot. Not going to worry too much about the positioning because obviously it's very out of focus. So, things that we can do to adjust. Well, it's probably mostly due to a misalignment of our objective lens. And I imagine that we need to get a little bit closer. So I'm just moving the objective lens a little bit closer to our sample until our dot comes into focus. Make sure that this is on the right side. Actually, no, it's not. We need to get this a little bit closer to our objective. I had flipped this around. We want the dot to be as close to our objective as possible because that's sort of where I measured my focal plane from. Yeah, and we'll back up a bit. We'll make sure that this dot is still visible. Small adjustments to our objective lens. That looks pretty sharp to me. And now I will play around a little bit with our camera positioning just to make sure that it's nice and exactly one focal length away. With an infinity corrected system, as I sort of showed you in the last lab, it's very forgiving for even slight misalignments because you do have this space in between these two lenses that is effectively collimated. You don't have an infinite distance between these two that you can play with, but even if it's a few millimeters off, you do get so very forgiving, very little reduction in image quality. So this is again one of the many advantages to building a Köhler microscope. Additionally, you get a much more even illumination because we do have light that is being delivered by our condenser into a nice, perfectly evenly illuminated plane that is, again, very forgiving depending on where you put your sample, even if you're a little bit further back or further forward. Because the light is focused, it is nice and even no matter where the sample is placed. All right, so this is a roughly aligned Köhler infinity corrected digital microscope. What we're going to be doing now is messing with this, this iris that we worked with in the last lab. Now you'll remember in the last lab there were certain planes that I could put the iris in where it would illuminate, where it would change the illumination on the sample either by changing the area of the sample that was illuminated or simply changing the total illumination that the sample received. And that is going to be, respectively, a plane that is conjugate to our image plane, and then, or to our sample plane, which is conjugate to our image plane, or one that is reciprocal, right? So perfectly out of focus. So let's try to sample. Let's just see where we are now. So if I start closing my iris where I am now, let's see what happens to our image plane. Well, it doesn't. You don't appear to be getting an image of our teeth on our image plane. You can see that it does become dimmer. The camera does sort of auto adjust for the total light that hits it, but I can put a small little piece of cloth here, and you can see that as I open it up, it is quite a bit brighter and dimmer. So we're definitely not finding a plane that's conjugate to our sample. So we need to sort of try to find that. So let's try to move this back a bit, and I will partially close my iris. See if we can find a plane where our teeth appear on our sample. So it is a little bit difficult to see on the camera itself, and that is simply because the camera is such a small field of view. So what we can do instead is I will slide this camera back, and then we will I'll mount sort of a small piece of paper here. Now hopefully you can see that at this point I form an image of my iris at roughly where my sample plane is, right? So you can probably even roughly right about there see the dot that is in our plane, and because the iris is large, it can be a little bit hard to see on just the small area that our chip was imaging, but you can see a roughly in focus image of our iris on our sample. It's not perfectly in focus, and that is because we're not building sort of what we call a 3-lens Köhler microscope. I'm just going to go ahead and slide this back until our dot comes back into focus. So in a proper 3-lens Köhler microscope, the field stop, which is the plane that we're sort of finding, would be at the back focal plane of our collector lens. We can't quite get there, but we can get close enough. So we can really sort of narrow down the total amount of our sample that is illuminated. And it might be, so to my eye, I can see that as I open and close this iris, I am also opening and closing the amount of area of my sample that is illuminated. So this is useful for simply controlling the total amount of illumination that hits our sample. But when you are dealing with a system where you only have one iris, it's actually more useful to have the iris mounted at what is known as the aperture stop. So instead of being conjugate to our sample plane and our image plane, we're going to try to find a plane in here that is reciprocal to it. Now, from your readings and class, you should know that the focal planes of a lens are reciprocal to each other. The back focal plane is reciprocal to the front focal plane. And we have a condenser whose front focal plane is mounted at the sample. So we should intuit that the back focal plane of the condenser, which is again going to be roughly one focal length away from our condenser, to make sure that we measure that, it should be about 35 millimeters. A little bit closer. There. This should be reciprocal. So as I narrow down my my iris, I can actually look at my sample plane, and I notice that I'm not getting sort of an image of the iris or of the teeth of the iris on the plane, on the sample itself. And again, it's probably a little hard to see on the camera because you don't get a full view of the field. But I will move my camera back again, and we will look at what my image plane looks like just by eye with a lot lower resolution. So let's go ahead and open up our. Just going to open that up. So as I close down my iris, you can see that you don't see an image of the teeth appear as you did before. You just see a dimming of that light. And then a pattern should emerge that you might recognize. And this is the LED. Because again, you should know that this is roughly the undiffracted light. We're not letting in all of the high angle illumination rays, only the very low angle illumination rays. So you can begin to see sort of the formation of this, of the LED on it. So let's narrow this down, and we will move this back and forth. It's pretty forgiving. So that's about right. We'll go ahead and move our camera back. Focus on our little dot there. All right. So we've found the field stop, and we have found the aperture stop of our two lens infinity corrected Köhler microscope. Now let's start thinking about, all right, what is the numerical aperture of this system? So in the lab, it asks you to calculate what is the numerical aperture if the condenser is fully closed, if the aperture stop is fully closed. Now I've already taken some measurements before I started assembling this, but you'll know from the last lab the iris doesn't really close all the way. This isn't a perfectly blocking iris. It closes down to a minimum diameter of about 1 millimeter. That's why we're getting just a little bit of light let through. So if we know that the minimum diameter of illumination light that we're letting through at this back focal plane is 1 millimeter, and that is mounted a focal length away from our condenser, then using the formula for numerical aperture and a little bit of trigonometry, you should be able to draw some triangles and figure out exactly what is the radius of light that is entering our condenser lens, and then how far away from that condenser lens is it being focused to, right? So again, 35 millimeters to our sample, you have 1 millimeter of light that's being illuminated. Calculate what the numerical aperture of this system is when our iris is closed. Go ahead and pause the video to run through those calculations. And once you've done that, we will now calculate what is the numerical aperture of our system when the field, or when the aperture stop is fully open. Now, what is limiting the amount of light in this case? Is it the aperture? Well, I can move this a little bit closer to my condenser, and I give that a look. It looks like the aperture is larger when it's fully open than the condenser itself. And I can sort of confirm that if I do some quick little measurements. This aperture opens up to a maximum of about 32 millimeters from edge to edge, but our condenser lens is only 28 millimeters in diameter. That's sort of the clear aperture that allows it to that allows light to go through after it's been mounted. So when this is fully open, I'm no longer restricting the amount of light that is maximally incident on our condenser. So then you don't need to include the iris in this second calculation at all. You should simply be able to think about, okay, what is the diameter of this lens? Again, it's going to be 28 millimeters. And then what is the focal length of this lens? 35 millimeters. And you should be able to calculate the numerical aperture of this condenser lens as when this iris is fully open. It does ask you what is the numerical aperture of the entire system. You can also run through calculations on what is the sort of numerical aperture of our objective lens. And if you do just a quick measurement, you can see that our objective lens to my eye is around 25 to 28 millimeters in diameter, but its focal length is much shorter than that of the condenser, as you can sort of see in the setup, because the objective lens is mounted much closer to our sample than our condenser is. Again, this focal length is only about 20 millimeters. So what is restricting the numerical aperture of this illumination system right now is not the objective lens, it is the condenser. So you should only need the diameter and the focal length of the condenser in this calculation to calculate what is the numerical aperture. All right, so you should go ahead and run through that. And that is everything that the lab asks you to fill out, but it's not super interesting. Just look at dots. So since we have now completed the assembly of a proper Köhler infinity corrected microscope, we could look at more interesting things. I have mounted a few pieces of my hair on a slide. Oop, and I'm gonna mount that the proper way, just so we can sort of see what this looks like under Brightfield illumination. There we are. So my hair is dirty blonde, not sort of the most pigmented of hair colors. But you can see that in this setup, it does look relatively dark compared to the background, because that's, and you should note that is what Brightfield really tells us, is simply the absorption and scattering properties of our sample, right? The hair in this case looks quite a bit darker than the background because compared to the water, even the small amount of melanin in my hair absorbs quite a bit of that light and looks pretty dark compared to everything else. And you can check out maybe another hair, see if I can find another one in here. Oop. Yeah, so the edges will look a little bit darker than the middle. I mean, my hair is, partially transparent. A lot of crystalline protein structures in there. You get a little bit of, you get more absorption at the edges because it will sort of refract light away from our objective lens. But overall, much darker than the background. So that is what hair looks like under a Brightfield microscope. I also went by the pond near my house and I collected a little bit of water, sealed it up. Let's see if we can see anything interesting in there. So we got to make sure we get this aligned. Oh, I always put this in the wrong way. You want to make sure that your cover glass, which is the thinner part of your sample mounting, is closer to your objective. That will minimize the aberrations in your sample. All right, so see if we can visualize anything in here. Oh yeah. So just some pond scum. There were some smaller creatures moving around in here earlier. Little Daphnia and Euglena. You can see a couple little swimmy guys. Pond water is always very interesting. There's lots of slowly decomposing matter. It attracts a lot of microbes, which then attracts a lot of protists. You can find maybe a bug or two sometimes. It's especially, they tend to be very active during the summer. There's a lot of light and heat. A lot of the more photosynthetic organisms like to colonize ponds. Typically the murkier the better. Murky means that there are things living in it. So, again, you do see, a negative contrast here, where your background is very bright and your sample is typically very dark because it acts as effectively an absorption filter for the illumination light as it passes through the sample, and then by the time it gets to the image plane, you're looking at a decreased intensity of light where there is something in your image plane and a brighter background where there's not anything. So very good for things that absorb light. Pond samples are typically pretty good because you have lots of things that have chlorophyll in them. Chlorophyll, as you know, is very green. Absorbs pretty much everything that's not green light. So it acts as a color filter effectively. And you can even see with some of the scum, there's probably a bit of a green tinge to it. And even if I jack up the intensity of my illumination light, things get a little bit washed out, but still are quite a bit darker than the background. So, you know, it's okay if you find this boring, because in my opinion, Brightfield is one of the more boring aspects of microscopy, as you have learned in class. There's other things that we can do, like introduce polarizing filters or an annulus to create dark field microscopy. And so that's what we're actually going to be doing in the next lab, is adding in an annulus to make dark field, turn this bright field color microscope into a dark field one. And so with that, that concludes lab 5. Like we said, we'll pick up with lab 6, where we'll be working with dark field microscopy.